Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Upstream Process Development for Better Yield and Product Quality. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speaker. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speaker throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Aragen Life Sciences who developed the content for this presentation. Aragen Life Sciences is a leading R&D and manufacturing solutions provider for the life sciences industries worldwide. It offers end-to-end integrated or standalone solutions for small and large molecules. Established in 2001, the company operates through a network of sites located in India and Morgan Hill, California, USA, with a team of 3,700 plus scientists and more than 450 PhDs. Its expertise and experience have enabled over 400 customers to advance their research programs from discovery through commercialization. Aragen's innovative mindset, infrastructure, and flexible business models have enabled them to serve large pharma, biotech, agrochemical, animal health, and performance chemical industries globally. Now, it's my pleasure to introduce you to our speaker for today's webinar, and there he is, Dr. Ali Abghari, Senior Scientist at Aragen. Dr. Ali Abghari is working for Aragen as a senior scientist with 12 years of experience in upstream bioprocess development, growing cells in the laboratory and pilot setting for the production of large molecules. He holds a PhD in biological engineering and is currently involved in the cell culture process development. Now, it's my pleasure to pass the mic and the controls over to Ali. So, Ali, when you're ready, you may begin.
Hello, Sonia. Can you hear me first?
We can hear you.
Perfect. Hello, bioprocess developers, researchers, and biopharma leaders. Thank you for joining this webinar session. In this webinar, we are focusing on the upstream bioprocess development for better yield and quality. Here is the overview of today's webinar. We will explore tools and methodologies for upstream process development around CHO cell platform. Also information related to the process scale up and process scale down, as well as major challenges and strategies will be discussed. In this session, I will refer to Chinese hamster ovary as CHO cell. The engineered CHO cell system is the major mammalian workhorse for the industrial production of therapeutic proteins. About 70% of therapeutic proteins is produced by this platform. The top advantages of this platform include its adaptability to various culture conditions and its high propensity for genetic changes. But advantages are not limited to this. On the left side, other benefits to include are the capability of CHO cells to grow in defined medium and its capability to achieve medium to high cell density and product yield. It is possible to obtain high product quality thanks to the ability of these cells to properly fold proteins. And also this platform has a capability for the generation of human-like glycosylation pattern. And finally, they have good ability to secrete proteins. But as compared to bacteria and yeast, this platform may suffer from the relatively slower growth rate and longer production time. Also, the overall cost is relatively high. And finally, there is a chance of contamination with prions and viruses when we are using these cells. Achieving higher yield requires productive cell line and optimal process conditions, including upstream and downstream processes. We also leverage analytical techniques for monitoring and controlling the process. I'm going to expand further on the workflow of generating stable cell line in the next slide. The process of developing cell line starts with the transfection of selected host cell with exogenous DNA. That is followed by generation of mini pools and selection of top productive pools. The selected pools undergo single cell cloning or single cell printing to generate clones, which go through further expansion to obtain the lead clones. Once we have the lead clones, we perform preliminary selection of medium and feed using shake flask studies. Sonia, if you don't mind, can you please take care of the first poll question for our audience?
Certainly I will. Okay, audience members, we're going to launch our first poll question, and there it is. You should see it in front of you. Now, I'll give you about 45 seconds to complete this simple poll question. What is the estimated market value of Chinese hamster ovary cells, Cho? Is it 1,000 to 1 million? Or is it 1 million to 100 million? Or is it 100 million to 1 billion? Or is it 1 billion to 100 billion? So please go ahead and select your answer, and then we'll have Ali give us the correct answer. Okay, go ahead and make your selection now. It looks like we have half of our audience, Ali. I'm just going to wait a few more seconds as it looks like other people are just making their decision now. Okay, that looks like it's the majority. Oh, perfect. Thank you so much, everyone, for participating in our first poll question. Now, I'm going to close the polls and let's take a look to see who chose what. So in our audience, 0% chose the 1,000 to 1 million. 27% of our audience said 1 million to 100 million, while 31% said 100 million to 1 billion. And then the majority of our audience chose, 42% that is, 1 billion to 100 billion. Wow. Okay, so the correct answer is what, Ali? And don't forget to put on your microphone, Ali. Can you put your microphone on? We can't hear you.
Perfect. Can you hear me now?
Yes, we can hear you now. Thank you.
Beautiful. So I think 31% of the audience, they made a correct choice. The correct answer that we have is C. According to this reference, Cho market valued at about $280 million in 2022.
Wow.
And that is expected to reach about $688 million by 2030. And thank you for participating in this round of poll question. So now I'm going to continue and go to the next slide. Implementation of innovative technologies for producing quality products with higher yield is critical. Examples of these strategies are presented here. We use single-use bioreactor to shorten the lead time and minimize the chance of contamination. We take the advantage of high throughput system to conduct design of experiments investigation under controlled conditions. We also implement quality by design approach to maintain steady workflow, resulting in the products that meet the predetermined quality standards. I'm going to provide more information regarding quality by design in the next slide. Quality by design is building quality into process. It is a systematic risk-based approach whose application will result in the following outcomes: product quality, regulatory adherence, cost saving, and expedited development. Design space is established based on complex relationship between process, quality, and efficacy. We place we aim to define process boundaries beyond which process fails to produce acceptable products. We also perform process control based on quality risk management, which I'm going to touch upon shortly in the next slide. Quality risk management. For quality risk management, we start by defining the scope of process. We use target quality profile to draft the design of process. That is followed by risk assessment, where we identify risks caused by process inputs, and I will elaborate on inputs in upcoming slides. Then we apply dynamic response and risk-reducing strategies for controlling the identified risks throughout the process. And finally, we review events and documents, and we try to document them for further improvement of the process. This slide provides more details on tools and steps we typically use. We start with process knowledge, including inputs and their impacts. We use tools such as failure mode and effects analysis for prioritizing inputs based on the level and frequency of risks caused by them. Then we proceed with screening stage, where we investigate the impacts from inputs with moderate to high levels of risk. Subsequently, we fine-tune the parameters to achieve favorable objectives. And finally, we monitor and control important inputs to maintain the output within the regulatory space throughout the product life cycle. Let me remind all of us that design space that we established originally is going to be constantly challenged with new data that we generate. As I mentioned before, I'm going to talk more about process inputs. The process inputs are screened in terms of effect on process outputs. The inputs can be related to seed preparation, medium, and process. We classify inputs into key process parameters, or KPP, and critical process parameters, or CPP. KPP only affect key process indicators, or KPI. But critical process parameters affects both KPI and critical quality attributes. Let me provide you with an example. Feeding regime. Feeding regime can be an example of CPP, whereas inoculation density can be an example of KPP. I will come back to this example when I'm presenting the process outputs in the next slide. Generally, critical process parameters should have been prioritized based on the level and frequency of risks that they might cause. Now I'm going to the next slide to present more information to all of you about process outputs that can be impacted by process inputs. Let's take a closer look into process outputs, which we want to categorize them into KPI, key process indicators, and CQA, or critical quality attributes. Some examples of these outputs are shown on this slide. You can see KPI examples and you can see critical quality attribute examples here. I want to refer back to previous example, the fitting regime. The fitting regime as the potential CPP, or critical process parameter, can affect osmolality, which is a KPI. It can also impact glycan profile, which is a CQA. We carefully evaluate the profile of process outputs, and our goals are to establish a robust process with a consistent quality. Sometimes, in reality, it is not simple to simultaneously achieve these two goals. So we rank our critical quality attributes and we try to find a sweet spot where both goals, robust process and consistent quality, are more or less met. Sonia, now I need your help again for the second poll question.
I'm right here. Here we go, audience members. I'm launching our second poll question. Okay, so here's a question. What are the top 2 monoclonal antibodies making up the majority of the approved mammalian cells-based antibodies on the market? Is it B, bispecific, 2, fully human, murine, chimeric, or humanized? So you have the options below as we're asking for which top 2 monoclonal antibodies. Is it A, 1 and 2, B, 3 and 5, or is it C, 1 and 4, or D, 2 and 5, fully human, humanized? Once again, that question is, what are the top 2 monoclonal antibodies making up the majority of the approved mammalian cells-based antibodies on the market? So we have the options there for you to choose from. And then I will close the polls, and then we'll look at the results to see what our audience chose, and then we'll get right back to Ali with the correct result. Okay, perfect. That looks like it is the majority of our audience. So Ali, I'm now going to close the polls and let's see what our audience chose. Okay, so in our audience, we have 7% that said the 1 and 2 combination. Then 17% said 3 and 5, while 31% of our audience said 1 and 4, and then 45% of our audience chose 2 and 5, human, fully human and humanized. So Ali, what is the correct answer?
And it looks like the majority, 45%, made the correct choice. Answer D is correct. So
2 and 5.
yes. So basically, according to this reference, humanized monoclonal antibody and fully human monoclonal antibody account for 46% and 36% of approved antibodies from the year 1987 to the year 2021.
Wow.
Great job.
Okay, back to you.
Perfect. Thank you, Sonia, for your wonderful support. So now design of experiments. Design of experiments come into place for the investigation of inputs with moderate to high levels of risk. With the help of design of experiment, we aim to determine how selected inputs affect process outputs. We come up with a statistical model as a predictive tool to relate critical process parameters to the process outputs, KPI, or critical quality attributes. As we move forward with the process development and we generate more data, we constantly try to refine our models. Which statistical design, which statistical method we want to choose and use, it all depends on what are the questions we need to address. What level of throughput can we achieve in our experiments? Which phase of process development are we currently in? For instance, we can use CCD, or central composite design, a common design of response surface methodology, to determine acceptable range as well as worst and best case scenarios for our process. Combination of high throughput experimentation and machine learning can also be employed when we are dealing with large and complex data sets. Speaking of high throughput, let's take a closer look at it. With the help of high throughput bioreactor system, we can generate richer data in a shorter time frame. Amber 250 system is an example of high throughput bioreactor system, which can be used as a scaled down model to simulate the commercial process performance. And also it can help with meeting regulatory requirements. Our Amber 250 platform enables control of up to 24 reactors, faster implementation of quality by design, cost-effective process scale up and scale down. And finally, it can help with the identification of process bottlenecks for re-engineering process steps or ingredients. Our Amber 250 platform is integrated with Vicell for inline cell counting. It is integrated with Flex2 for inline metabolite analysis, and with MODI statistical software package for data modeling. In addition to MODI, JUMP and Design Expert can also be used for drafting different plots and determining robust and optimal setups. Sonia, I'm gonna have your support one more time for taking care of the third poll question.
Okay, and here we go, everyone. Our final poll question for this webinar. And the question is, which one of the following combinations represent the scale-dependent parameters? So you remember you have a couple of options below you can choose from. Is it temperature, pH, and medium composition? Or is it agitation, rate, and Sparger rate? Is it DO, temperature, and agitation rate? Or is it agitation rate, working volume, and DO? So please go ahead and make your selection now, please. Okay, everyone's a little slow getting on this one here. Once again, it's which one of the following combinations represent the scale-dependent parameters. So please go ahead and make your selection, and then I will close the polls and pass it over to Ali to give us the results. Okay, that looks like it is the majority of our audience. I'll leave it open for a few more seconds, and then I will close the polls and we'll take a look at the results to see what our audience chose, and then go to Ali for the correct response. Okay, I'm going to close the polls now. Thank you, everyone, for participating in our final poll for this webinar. Okay, so here are the results, Ali. In our audience, 15% said temperature, pH, and medium composition, while 31% said agitation rate and Sparger rate. Then we have 4% that said DO, temperature and agitation rate, and then 50% of our audience chose agitation rate, working volume, and DO. So the question is now, Ali, what is the correct answer?
Sure, I'm very excited by the results. I see a lack of diversity in results.
Yeah.
The correct answer that I'm going to share with everyone is going to be answer B.
B. Oh.
Yes. And the temperature, dissolved oxygen, medium composition, and pH, they are typically maintained constant as scale independent parameter during the bioreactor scale up or scale down. So the answer B, agitation rate and Sparger rate, they are considered as scale dependent parameter.
Oh.
Yes,
Good to know.
thank
Thank
you.
you.
Thank you, Sonia, for your support, and thank you for the participation from audience. So now I'm going to continue. For a scaling up or a scaling down, we collect historic data from large to small scales. We consider similarities, demand, and limiting factors across scales to select and test scaling criteria. Examples of these criteria are all presented here. These are the typical criteria. After employing the selected criterion, or in some cases, combination of criteria, we look into the profile of outputs, KPI and CQA. By looking into graphical or statistical comparison, we verify equivalency, comparability, or differences among outputs across scales. We verify equivalency, comparability, and differences in order to determine how reliable our model is. We finally choose the scaling model, which allows for matched process performance. And I want to go through this process step by step in the next slide. We start by evaluating the inherent properties of our bioreactors, including geometry, aspect ratio, and control capabilities. Then we identify scale-dependent and scale-independent parameters. We consider critical process parameters and we investigate scaling models, including oxygen transfer rate or volumetric power input across scales. We overlay the output data to identify regions of overlap or discrepancy. The acceptable ranges are considered and principal components are identified. We demonstrate if the selected model is capable of simulating the process performance at a different scale. During this process, we always remain vigilant for challenges related to at-scale equipment. I will discuss common problems with the scaling in the next slide. Common challenges encountered during the scaling process includes maintenance of consistent quality across scales, shear generated by mechanical steering, aeration, or foam formation, and finally, insufficient provision of oxygen and buildup of excessive carbon dioxide can be a challenge to face and to deal with. To conclude this webinar, let's take a look at the key strategies for scaling the process. We fine-tune the composition and rate of spark gas. Sometimes we take the advantage of separate sparger elements for matching the oxygen and carbon dioxide profile. We aim to establish balance between agitation and aeration. We do this together with proper combination of sparger and impeller for enhanced mass transfer and minimal shear stress. Computational fluid dynamics, or CFD, can also be used to get further insight into the flow pattern for matching the mass transfer and stress heterogeneity. To deal with scale effects, we readjust the carbon dioxide flow in some cases, and in some other cases, we readjust the DO set point. Finally, we wisely select the placement of addition ports to optimize efficiency. In closing, I'd like to thank each of you for joining us today. And if you have any questions or comments, please feel free to reach out. Stay connected, inspired, and I'm looking forward to your questions. Sonia, I think you are on mute.
Am on mute. I keep on clicking, but it didn't work. Now we're here. Okay.
Yeah.
I just want to say thank you very much, Ali, for that very intriguing presentation. I hope everyone enjoyed it. Now we're going to begin the Q&A portion of the webinar. So I invite Ali to put on his webcam now, and I'll invite the audience to continue to send in their questions while you were speaking, Ali. I did receive some questions, so let's begin with that. Ali, you can put on your webcam there.
Sure.
And I'll start with this
I'm
first
working
question.
on that. Beautiful.
There we go. There he is, everyone. There is Ali. Okay. All right. So, one audience member just sent this question in. What are the key factors driving the future direction of CHO cell culture process development? What do you
That's
think?
a very good question.
Hmm.
There are many different aspects to consider when we want to answer this question, but I would go for some new technologies, including CRISPR technology for better genetic engineering in CHO cells. Or there are some cases that people they collect omic data for better pathway engineering. These two factors definitely are going to receive a high rank. But in addition to them, continuous perfusion process, especially on the side of upstream, is going to be play a huge role in the future to intensify the process, increase the facility productivity. And the other factor that I would like to mention would be process analytical technologies. Those technologies can be taken advantage. We can take the advantage of those technologies in order to set up dynamic feeding, which is gonna improve the quality of the process. And machine learning. Machine learning can be another factor that is going to play a more important role in the future, not only for optimizing the medium, but also for better design of experiment for the identification of more critical process parameters. These are the factors that I can mention to answer or to address this valuable question.
Okay, thank you. All right, let's go to our next question. Let's see here. This audience member is asking, can you please discuss the single cell cloning in more detail, please?
Sure. For single cell cloning, when we are developing cell art, basically the new cell line, we start with the transfection, as I mentioned before, and then we generate a lot of mini pools. With those mini pools, we screen them for the productivity, how much protein they are producing, and for their growth behavior, how they are growing. Once we narrow down our long list to a short list of top pool candidates, we come and we perform a single cell cloning or single cell printing on them in order to go from pools, which is a mixed culture, to separate clones, which I would say in terms of cellular machinery, like we are going to deal with purified clone, like one single clone. And that's very important for us because then we are going to build other steps on those selected clones and see which one of those clones are more productive. As I mentioned on my slide, we pick up the lead clones, and once we have the lead clones after single cell printing, we try to develop medium feed around it with some preliminary shake flash studies, and then we go for bioreactor cell culture or amber cell culture in order to optimize the process around the lead clones.
Okay, thank you, Ali. I'm going to jump to this next question here. This audience member is asking, are there any factors directly affecting the characteristics or potency of the product?
So yeah, there are a lot of factors that they can affect the characteristics of our product. Some of those factors, I think I mentioned on one of my slides, that they come from the way that we set up the medium, the way that we set up the process, a lot of different process parameters. They can affect the quality of the product. For example, sometimes we fine tune the dissolved oxygen. Sometimes we fine tune our feeding regime, or sometimes we try to uh, go for a specific temperature shift in order to get better product with higher quality out of that process. So yes, we play with different parameters, medium, process parameters, and sometimes we do our best in order to come up with the best clone, with the best cell line, which is the most productive one, and in terms of productivity and in terms of growth, and we
Mhm.
try to get the best product out of it.
Okay, perfect. Thank you, Ali. All right, it looks like we have about a couple more questions here. So a reminder to our audience, you still can continue to send in your questions using that questions chat box, and we'll try to address your questions during the time we have together. Now I'm going to go to our next question. This audience member is asking, what are the major strategies for controlling glycosylation patterns? That's a good one.
That's a very valuable question because
Mhm,
glycosylation pattern can directly impact the safety and efficacy of the product. And we have different strategies for that. Some of them we definitely do it in-house, some of them maybe not. But we can take the advantage of combination of genetic engineering. We can
mhm,
supplement our medium with different components in some cases, and in some cases we bring our major efforts into the process. For example, there are cases where agitation rate or temperature or dissolved oxygen, it impacts the glycosylation pattern. Or we have seen cases that osmolality, which is impacted by feeding regime, can also play a role. So by
mhm,
fine tuning the feeding regime, we can sometimes control or manage the level of osmolality, which is going to impact the glycosylation pattern. So
mhm.
it's like it's not a single strategy; it's a combination of strategies that we try to take the advantage of, and we all try to find the best combination for each process and for each cell line.
Okay. All right. Thank you for that
Good
very
question.
thorough answer. Yeah. Okay, let's jump to our next question. What are the major strategies for reducing waste byproduct accumulation? Another great question.
That is true. Another great question
Hmm.
because generation of byproducts like ammonia, lactic acid can be problematic in different ways. But again, in order to deal with that, it's like a combination of strategies that we usually put together to deal with that. I can name some of those strategies that how we try to deal with that. We can play with process parameters like temperature or like glucose level or dissolve oxygen and try to fine tune them in order to get the minimum level of generation of byproducts. But in addition to that, feeding regime, again, shifting from bolus feeding to continuous feeding, in some cases, it can also help with that. And on top of that, strain engineering, like more specifically, pathway engineering can also, I have seen some cases that with pathway engineering, people, they try to manage that or control the level of generation of byproducts. So again, combination of strategies are going to be basically used in order to uh, minimize the level of generation of ammonia or lactic acid, which are considered as byproducts.
Okay, thank you for that. I'm just so busy getting all the questions that are coming in from our audience. They're coming in fast and quickly here. All right, so let's jump to the next question here. Okay, when or how is the stability of the new cell line verified?
That's a good question. So when we come to our lead colons, that's the time that we try to make sure that the colons that we have came up with and we are going to develop process around them, they are going to stay stable, not only during the time that we are investigating them, but also during the time that we are using at manufacturing scale. So what we usually do, we for those top lead columns, we try to send them through stability test, which includes multiple rounds of passaging for 2 months or sometime a little bit more than that. And during that I call it incubation time. We try to monitor the productivity and the growth behavior of the cell to make sure that it's gonna show a consistent growth, consistent basically growth, and also in terms of the productivity, it's gonna stay consistent. And there are some stresses that sometimes those cells they are gonna deal with, for example, freezing, thawing, and we always try to be vigilant about the behavior of the clone after thawing. We want to make sure that if cells undergo some stresses, it's not going to lose its capability for growing in an acceptable manner and for producing again in an appropriate manner.
All right. Thank you, Ali. All right, let me go to the next question here. Okay, this one here is a little lengthy. Um, in the earlier question, you indicated DO is kept constant across scales, but in the last slide, you gave the option of adjusting DO to control CO2. Can you elaborate?
Absolutely, that's a very good question.
Mhm.
We play with DO and we directly measure that, but something more important would be the concentration of the soluble oxygen in our culture, in our medium. And when we scale up or when we scale down, we want to make sure that the concentration of oxygen, soluble oxygen, is kept constant.
Mhm.
And if we keep the dissolved oxygen constant at different scales, it doesn't necessarily mean that the concentration of soluble oxygen is the same across scales. Why? Because of the way that we do the dissolved oxygen calibration might be different. The way that we do it with bench scale bioreactor, or the way we do it with pilot scale bioreactor or manufacturing scale bioreactor. So that calibration might be different. Besides that, there are different parameters that they are maybe true for, they are very specific to a scale. Let me put it this way. Like for a small scale, for example, we don't have back pressure. But with large scale, maybe we have back pressure, or let's say hydrostatic pressure. And that pressure can also impact the solubility of oxygen in our culture, in our medium. So at the end, what matters is like we set up our process at a specific DO, which allows us to match concentration of soluble oxygen across scales. That is why I said that. Yes, DO set point, we play with that, we directly measure that, but when it comes to going from small to very, very large scale manufacturing scale, we always need to pay close attention to the level of soluble oxygen that we want to basically provide, we want to have in order to match them possibly across scales as much as possible. Good
Okay,
question.
thank you. Okay, thank you for that. Okay, I'll go to the next question here. Can temperature shift be used in commercial production?
That's a very valuable question. Definitely, there are ways to do that with a larger scale. However, something that we always should keep in mind that by lowering the temperature, we might be able to solve some issues that we are really dealing with. However, by lowering the temperature, we might elongate the process duration. So there should be sort of so there is going to be a trade-off, and we need to be aware of that. By
Mhm.
doing a very brief techno-economic analysis, we should be able to see how how much cost is gonna basically additional cost is gonna bring to the process if we want to lower the temperature to a specific limit and deal with longer process. And from that additional time that we bring to the process, how much additional productivity or maybe titer or maybe yield we are gonna get. Is it gonna justify that or no? For instance, if we lower the temperature and as a result of that, we elongate the process for more days, but because of those with those additional days, we
Mhm.
don't get that much additional of product, then maybe economically it doesn't make sense to do that. So we always have to take all of them into account, and the temperature that we want to pick up for implementing temperature shift, that also matters.
Mhm.
Sometimes we shift from 37 degrees to 30 degrees, sometimes 34. And so it really depends on the preliminary data that we
Mhm.
achieve, how much of improvement we can bring to the process by implementing that temperature shift strategy. Good
Okay.
question.
All right. Well, thank you. I think that's all the questions that have come in right now. So I just want to say thank you very much, Ali, for answering those questions. We have reached the end of the Q&A portion of the webinar. For those of you who are interested in hearing more from Aragen Life Sciences, and who wouldn't be, you can register for their next webinar, Accelerating Vaccines to Clinical Trials, a toolkit for efficacy, safety, and bioanalytical studies, scheduled for Wednesday, November the 15th. And I'm going to send you that link in your chat box right now. So please go ahead and register for that very exciting webinar. And there you go. Everyone should have it. Now, if we could not attend to all your questions, the team at Aragen Life Sciences will follow up with you after this presentation. And if you have any further questions, please direct them to the email address that you see there on your screen, info@aragen.com. Thank you everyone for participating in today's webinar. You will be receiving a follow-up email from Xtalks with access to the recorded archive for this event. Additionally, there is a link to view the recording of this event, which you can also share with your colleagues once they register for the recording here as well. And a survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve on our further webinars. Now, please join us in thanking Ali for that very insightful presentation. We hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at X Talks, we thank you for joining us. I'm Sonia Hunt. Until next time, please take care and bye for now. Bye, everyone. Thank you for joining us.